Thank you very much. Good morning / afternoon. Thank you for having me here. First time, fall in love with CannMed. Thank you for still being here. We're closing to lunch. I often hear people saying that nutraceuticals and cosmeceuticals are the lower hanging fruits compared to medicine, to drug. Is that true? Absolutely. If we talk about the regulation, we talk about the requirement for the approval. But I do still believe that any good products, any good cosmetic products, dietary supplement products should be supported by science, rigorous research, especially with the ones that has functional claims. So I want to use this opportunity to share with you the research experience we have. And I want to share with you the promising data, the excitement I have towards the development of nutraceutical or cosmeceutical ingredient based on non-cannabinoid compounds from hemp. All right, most of the fundamental basic research are done in the lab, in my lab at the University of Rhode Island. Some of them are supported by industry partners from different companies. And in addition to that, I also have a startup, very early stage startup called Ocean State Bioactives, OSB, to further develop potential cosmetic or nutraceutical products with different natural products. So in the past few years, the capacity at our university for cannabis education and biomedical research has been significantly increased. We have a group fascinating, awesome faculty members that we do together on the research of cannabis. In addition to that, I lead a research unit that is specifically focused on the potential application of natural products on their nutraceutical and cosmetic applications. Sometimes I take a step back thinking that this is not about cannabis, this is not about hemp, it's about any natural products. How do we use our platform? How do we rationally design good products? So comes to me, it always comes from two angles. The strategy to develop a natural products-based nutraceutical or cosmeceutical ingredient are always come from two angles. Of course, one is the chemistry, especially the phytoconstituents of natural products. And the second one is, of course, their biological activities. For hemp, I feel like this is very promising, very interesting, especially for dietary supplement and cosmetic industry. Why? Think about the hemp root. It's sometimes called waste products from the industry. I feel that's pity. I think it never need to be called as a waste product. It's a byproduct from the hemp or cannabis industry, but it has a great potential to be further developed into nutraceuticals or cosmeceuticals. Another example is the spent biomass. To a lot of people, it's just spent waste material. But as a natural product chemist, I think that one, the spent hemp material, still has a lot of bioactive compounds inside. For example, this is another side project. We have extracted a lot of bioactive compounds from spent hemp material, and especially for the phenolics and the polysaccharides. Those polysaccharides are closely associated with a lot of applications for dietary supplement. For example, it has been studied that polyphenol are good for the gut health, especially as prebiotics. In addition to that, we have set up a library of cannabinoids, and we use those cannabinoids for our bioassays. We set up an in-house panel, a panel of in-house assay that could evaluate the natural products in our library. The central of that library and also for that bioassay platform is inflammation. And from inflammation, we can derive to gut health, we can derive to skin health. For today's talk, I want to spend some time on the background of our research using CBD as a representative compound from cannabis. And I want to introduce you how we set up our biological evaluation systems for our further studies. Although the focus for this talk is on non-cannabinoids type of compound, but I do think CBD could be a very good chemical probe to study those the chemistry and the biological activities of this compound. Then I will move to the chemistry study of the hemp roots and their anti-inflammation activity, as well as their skin protective effect. So I do want to spend some time on the first part, introducing you the research we have done on CBD, on cannabinoids. The reason is that most of those studies are published. I won't go to too much details on the data, but I want to spend some time on how do we set up those assays? What are the rationale behind the publications? Share with you some of the workflow, some of the stories behind what you don't see from publications. The first step we had was that we set up a library of cannabinoids. This includes CBD and a few of those rare minor cannabinoids. We have about over 30 different minor cannabinoids. In addition to that, we did chemical modifications and we used synthetic chemistry to build up another separate library as the CBD derivatives. As you can see here, many of them are showing even better biological activities in different applications. We have studied all kinds of different biological activities of minor cannabinoids. This is, for us, is kind of like we understand those compounds better, and we understand the bioassay system better. Just like machine learning, you feed the machine with data, and they can improve getting better. Although we are not that fast, but every time we carry on a study using different natural products, different cannabinoids in different bioassays, the understanding of those bioassays and the chemistry get profound every time. So that's the one starting point we had. So here you can see, initially we test CBD in an cellular inflammation model. Specifically, this is an anti-inflammatory model. I will give you a little bit more details later. Then we pivoted from the monocyte, the immunocells, towards skin cells. In this case, we use skin keratinocyte as the cellular model. And we can kind of like shifted from the inflammation stress in the monocyte, human monocyte, towards the inflammation stress in skin models, and especially for inflammation-mediated cell death. In this case, is ferroptosis. And we also study another type of cell death in skin cells, which is ferroptosis. So this is the full cycle of the development, the evolution of our bioassays. Let me give you some of the details. So the first step is set up different bioassays. Those bioassays include one biochemical-based assays, could be enzymatic assay. Then, depends on different outcome or endpoints, we design cellular-based assays. This is very important because it helps us to understand the mechanisms of action of those cannabinoids better. Then depends on if it is necessary, we can move to animal-based studies. We can use different animal models for different diseases models. In parallel to that, I feel very excited that we also set up a new platform that utilizes more advanced technologies, such as proteomics or advanced binding assays to understand better the biological activity of those compounds. As you heard from the last talk, Hunter bring a very good point. When you do biological assays, especially for cell-based assays, you see the statistic, you see the stars, you know it's working or not. If it is the P less than 0.05. Yeah, sometimes it tells you if it's working or not, but you don't see the information, the dynamic change from the complexity of the biology behind the values, the p-values. So that's why we want to utilize more advanced technology to understand better the mechanism of action of those compounds. As you just heard from Dr. Goldenstein, that multi-omics is very rapidly adopted by the field, study the biological activity, the mechanism of action of cannabinoids. Here, similarly to metabolomics, we have a platform approach set up. It's about the proteomics to look at the landscape of the change of proteins from cells, from animal tissues, from human biological fluid samples to understand what's the impact of those cannabinoids on specific diseases models. In addition to that, we also set up this assay, this binding assay. So once you have a specific target from the proteomics, you want to validate if the compound can actually bind to that molecular target. In this case, we use a technique called SPR, surface plasma resonance, to confirm, to validate if the compound could directly bind to the protein, to the target. So we started the anti-inflammation assay with this model called inflammasome. So what is inflammasome and why it's important? Inflammasome, it's a group of proteins that could be assembled for the purpose of sensing infection stress. So in the normal status, there are just group of proteins. But when the human body sense the stress from infection, those proteins, they will assemble together and further activate the downstream protein sequences. For example, they can activate one protein called caspase-1, which is a very important enzyme that could further activate the secretion of pro-inflammatory cytokines, such as IL-1 beta. So the point is that if you through any natural products, you more or less see anti-inflammation activity, very general anti-inflammation activity. If you search a certain natural products in PubMed, in Google Scholar, you always see it has inflammation activity. But sometimes inflammation is not a bad thing. The root of all the diseases are chronic inflammation. And we found that inflammasome is more pathologically relevant to inflammation, to chronic inflammation. So we want to set up an inflammasome-specific model to evaluate our compound. So how do we do that? This is actually very straightforward. So in the general inflammation assay, often you do is that you expose the cells, monocyte, macrophage, to the inflammation stressors like lipopolysaccharide, LPS. Then you measure the outcome, you measure the biomarkers, inflammatory biomarkers. In this case, in addition to LPS, we add a secondary signal, which is known as nigrosin. So after the cells have exposure to LPS and nigrosin, is set up an inflammasome model. With this model, we can screen the library of our compounds. And I found that very interesting that we screened over 300 different natural products, not only cannabinoids, many different natural products. Only a few of them showing specific selectivity towards the inhibition of inflammasome. In this case, CBD is one of those most promising inhibitors for inflammasome. So those are some of the details of the study which are already published. So I won't go to too much, but basically at the non-toxic concentration to the cells, CBD was effectively reduce the production of IL-1 beta without affecting TNF alpha. That's how we can tell it's a very specific inflammasome inhibitor. So after that, we want to have more evidence to support the anti-inflammasome activity of CBD. So we further develop different assays to measure the receptor P2X7, which is very important mediator for inflammation. So we can say that using our different flow cytometric assays, we can show that CBD could effectively reduce the inflammation level in the THP1 cells. Then we shift to skin. One of the reason is that inflammation is actually very pathologically relevant to many of those skin diseases, acne, and even to aging. So that's why we want to say if that inflammation the anti-inflammatory activity of CBD could be translated into skin. So what we have is that we have different bioassays set up. We can say CBD could effectively reduce the cellular oxidative stress in the skin keratinocyte high-cut cells. And we use GenePlex assays to look at a panel of different genes that are relevant to inflammation. And very interestingly, we identified that caspase-1, the one activated IL-1 beta, was actually identified from this panel, and which was supported by our computational analysis. So furthermore, we tested the inhibition activity of CBD on caspase-1, and eventually we use SPR technique to directly measure the binding affinity between CBD and caspase-1, and all of those are published data. So caspase-1 is the one that closely mediates the process of Ferroptosis, which is an inflammation-driven type of cell death. And we know that skin is more prone to UV damage, which is closely associated with another newer type of programmed cell death called ferroptosis. So what's ferroptosis? It's a programmed cell death, and it's majorly mediated by two biomarkers. One is the iron level in the cells. Two is the lipid peroxidation in the cells. So our assay, we set up this assay, we expose keratinocyte cells to different inducers, including elastin, which is a ferroptosis inducer in this case. And we use this model to screen a whole library of different cannabinoids. CBD, again, very active, and that there are more minor cannabinoids that were more promising than CBD. So this is the data for CBD. It could effectively reduce the damage, the cell damage from elastin, and also reduce the two most important biomarkers, lipid peroxidation and the iron level in the cells. Furthermore, we use proteomics to take a look at the landscape of the protein change in this process, and we can understand better what kind of proteins are involved in this skin protective effect of CBD. And all of these are published data. If you have more questions, I would be more than happy to answer them. So after basic research, we still extended to kind of application research. If you talk about the dietary supplement, we in this case, we measure the stability of CBD in GI tract, and we also study the skin penetration capacity of CBD using artificial membrane-based assays, including PAMPA, Francis cell diffusion of those bioassays. All right, to the next part is the phytochemical investigation of hemp root. So this was very interesting if you read Dr. Russo's review paper on the cannabis root, which could be a traditional treatment and also a future treatment for pain and inflammation. So I was very interested in this part of this plant. Then I paid a visit to my collaborators in China. They're actually the largest producers in China for hemp. And when I visit them, I see just a massive amount of hemp root, dumped on the side of the road. And they do nothing about it. They call it waste products. And they either burn it or just landfill. So for me, as a natural product chemist, I feel pity because I know there are so many good goodies in this part of the plant. So what we did is a phytochemical evaluation of this part of plant, hemp roots. So you can say, I'm not going to every compound that we have isolated, but you can see apart from few cannabinoids, there's a large diverse of bioactive compounds in hemp roots. The chemotypes of those compounds are amazing. You have phenolic compounds, you have triterpenoids, you have phytosterols, you have fatty acids, but they are often overlooked in terms of drug development. But they could be very valuable for nutraceutical or cosmeceutical development. Just some highlight here is that some of those compounds are actually the first time that has been isolated from hemp or from hemp roots. So that's the chemistry we have done in this part. Then we further took all of those isolates into the inflammasome inhibition assay. We first did a screening assay to see their general anti-inflammation activity. Then we tested the cannabinoids from the roots and also the non-cannabinoids, which are showing activity in the screening assay, showing testing if they have specific anti-inflammatory activity. One of those compounds showing anti-inflammatory activity, which is not a cannabinoid type of compound. So we took one more step further, although this one has been published, but we did one more step of study. Because what we have missing is there are so many different possibilities to get the different type of compounds from hemp root. For example, here we use different extraction technology to further look at the chemistry, the phytochemistry constituents from hemp. This one we were specifically looking for alkaloids from hemp roots. Very interesting, this is some unpublished data. This is our preliminary result. So we have fractionated those extracts from hemp roots, and we further generate the fractions, the extracts that enriched in alkaloids. We have so far four fractions that has enriched in alkaloids. And you can see their anti-inflammation activity, 3 out of the 4 has very good anti-inflammation activity. So that was really triggered us. We really want to go deeper to see what kind of compounds are responsible for this anti-inflammation activity. But if you think about the alkaloids in hemp roots, you cannot forget about those compounds called cannabis. Those are the typical, very representative compounds from hemp roots, and their natural abundance in the root is very tiny. It's very hard to isolate those compounds from the roots. So now we have enriched a fraction that could have potentially this type of compounds. From the preliminary data, from the NMR, from the mass spec, HPLC, TLC data we suspect that there's one fraction of those alkaloid enriched fragments that is enriched in spermidine, which is the type of compound, spermidine, which also showing very strong anti-inflammatory activity in our bioassays. So our goal, our aim is to further look at what are the chemicals in this fraction that could be responsible for this anti-inflammation activity. And if there's a new compound or more representative compounds that could be further used as the standards for the future development of dietary supplement or nutraceuticals. Why the spermidine I feel so excited about that. Many research support that spermidine is a dietary compound could potentially affect the longevity of a human body, and this is supported by different type of models, animal models, and it's heavily associated with autophagy, which is also a promising strategy for longevity research. And you can see now there are already many this type of products you can buy from Amazon, and if you search spermidine type of supplements, you can find many of them. But what I feel pity is that those type of products, more often you don't see the standardization. You don't know what exactly the spermidine type of spermidine compounds are in the bottle. So there are so lack of research to support their potential application, their functional claims for those type of supplements. So I think that's something we can do more about this. Okay, one last part is the skin protective effect of compounds from hemp roots. So again, we go back to this process. In addition to isolate alkaloids from hemp roots, we'll also look at phenolic compounds. And we were able to detect, this was only a detectable level. So meaning that the natural abundance of this compound, cannflavin, is very minor in the hemp root. So we were able to detect it, and we did some biological evaluations here too. So what we have here, again, we set up a cellular model using a human keratinocyte, the skin cells, and we challenge the cells with different oxidative stressors. Then we extract the protein and analyze the proteomics to see what kind of biomarkers are related to this process. And this is a busy slide. I'm not going to the details, but our bioinformatics, they guided us to walk through all of those biomarkers and constructed different network to further explore the potential biotarget and the pathways in this process. And from this large amount of data, we predict that flavonoids could be an effective compound that could be used for reduce the damage for the oxidative stress in the skin cells. So what we did was that we get cannflavin A and B. We threw them into this cell-based assay for oxidative stress in the skin cells. And we did the biological assays. We were saying that at the non-toxic level, cannflavin A and B could both effectively reduce the damage of the cells from oxidative stress. It could reduce specific biomarkers that is related to oxidative stress in the skin cells. Last part, we also have the mechanism of mechanistic study to show that what could be the possibility of those compounds in terms of reducing oxidation and also reduce glycation in the skin cells. And this part has been published as well. So one last thought I want to share with you is that when we talk about development of a potential nutraceutical or a cosmeceutical, just like pharmaceuticals, CMC is the last barrier for the drug to get approved. You heard about that from the panel yesterday. So very similar. When you're thinking of develop a nutraceutical or cosmeceutical ingredient from natural products, you always need to think about, is it feasible? It is commercially possible. Even you have a lot of strong, rigorous data to support the function. In this case, for example, cannflavin A, we were able to isolate grams of cannflavin from hemp material. But this 30-gram cannflavin was isolated from over 600 pounds of the dry material. So it's almost impossible you can extract a fraction from the hemp or from the root and further use that for industrial scale up. It won't just be feasible. So what could be the alternatives that we can produce this type of compound? As you just heard the presentation before, synthetic pathway could be one route, could be one option to go. So in this case, OSB had a collaboration with contract research. So we now establish the organic synthesis of cannflavin A. Not only cannflavin A, cannflavins A, B, C, and isocannflavin B. So we were able to scale it up to gram quantity. In this case, we scaled it up, the production of those cannflavin in gram quantity, which will enable us to do further application research on the cosmetic or nutraceutical development. So this is what we have so far. Okay, just summarize here. We have looked into the chemistry of cannabinoids. We have looked into the chemistry of hemp roots. We found that this is amazing. You have a diverse group of bioactive compounds from hemp roots. And there are so many potential compounds, we haven't even discovered them. We don't know what are they. And I believe they do have the chance to further be developed into different nutraceuticals. And the data from the biological assay support that point. So I feel this is very promising in terms of further development. And that's exactly why I think we should not rush it into a products and sell the products. This has to be do more research about it, toxicity, safety profile, application research, formulation. There are tons of research we have to properly do in the right way to support its further application as dietary supplement or cosmetics. So that's my last slide. I know that I used all the time for the presentation, but I will be here all day long. So if you have any questions, feel free to come to me. I would love to have a discussion with you. Thank you so much.